and good morning all. I'm going to tell you about our task in MedAid, and that's the development and applications of the MedFish SNP array, which you've already heard a little bit about. I'm based at the Roslin Institute, which is part of the University of Edinburgh, but this was a collaborative study, and I'll mention the others involved later. So this is the outline of my talk. I'll give you a bit of background on the objectives of our work, what we did in terms of materials and methods, summarize the results, give you some conclusions, and then talk a little bit about the impacts and exploitation. Anna has covered some of this actually, but just as a brief reminder, so selective breeding to improve production traits has major potential to improve sea bass and sea bream production, including in the Mediterranean. And genomic selection is the state of the art in advanced breeding programs. It's really been a proven technology first in terrestrial livestock, and then now moving towards aquaculture, starting with Atlantic salmon, but moving now to many other species. The reason is it can result in markedly improved selection accuracy and genetic gain, as we've heard, and all doing so with reduced improved ability to control inbreeding, and therefore potentially higher genetic gain with reduced inbreeding. And genomic selection does require the application of or the collection of high density genetic markers across the genome of the species of interest. And one popular way to do that, as Anna mentioned, is a high density SNP array. These are the sort of workhorses of genomic selection for the majority of aquaculture species worldwide. So what we aim to do is to develop a combined species SNP array with 60K markers, a fairly standard marker density for these platforms. But we chose to produce a combined array because it was more cost effective to put sea bass and sea bream SNPs onto the same array. And we believed that 30K SNPs per species was more than enough for what we need. And I think we've hopefully seen in the project that that is a correct assumption. So all this work was a collaboration, so it was a really enjoyable collaboration both within MedAid and between MedAid and PerformFish, as Anna mentioned, the sister EU project of MedAid. Both projects contributed extensively to samples and sampling from throughout the Mediterranean, primarily farmed populations or breeding program populations, but also some wild populations of both sea bass and sea bream. And you can see there the reasonably good representation of the geography of the Mediterranean and a couple of Atlantic populations also. So our pipeline for work for large-scale discovery of SNPs in the two species. First, of course, we extracted the genomic DNA from the samples that we obtained. We then took a strategy of SNP discovery called pool sequencing, which is where we pooled the genomic DNA per population, had 2 pools per population, performed a lot of high throughput sequencing using the Illumina platform, then moved on to discover a big database of SNPs. From that list, we then did some filtering, which I'll talk about in a second, and ended up with our final list of SNPs for including on the SNP array. So our issue, if you like, was that we had very many SNPs to start with, dispersed all throughout the genome of both sea bass and sea bream. We had tens of millions of them, and we found that we needed to filter to have even coverage of the genome, accounting for the local nucleotide diversity. So we wanted to cover areas that had higher rates of recombination with a higher SNP density. And we also ensured that we had some overlap of our SNP platform with existing platforms that were developed in recent years, which was the iFremer or GeneC bass and bream SNP arrays. So the finalized design of the SNP array, as I said, contained 60,000 markers approximately. There was an average of about 16 to 17 kilobases between each marker on the genome. You can see there on the right the process and just the numbers in terms of the number of SNPs we started off with. The reason it's higher in sea bream is really just the depth of coverage was higher for the sequencing. I don't think it's necessarily reflecting a higher amount of variation in the sea bream genome, but that was the process we went through. And then this diagram shows the distribution of the SNPs across the genomes of sea bass and sea bream. And you can see in track A of this diagram is the chromosomes of the two species. In track B is the nucleotide diversity, which is a proxy of recombination rate at that particular point in the genome. And then track C is the distribution of SNPs on the MedFish SNP array. And you can see that the distribution of SNPs tracks quite nicely with the nucleotide diversity for both species, and we have a good coverage of all chromosomes. We then moved on to test the SNP array performance in diverse populations. Of course, some of this was also in collaboration with others. And we genotyped the samples that we used for the SNP discovery and categorized the SNPs according to their genotyping performance using some software provided by Thermo Fisher. What we found is that from all of our candidate SNPs that we provided and placed on the array, the top category we were looking for is high quality polymorphic SNPs, particularly polymorphic high resolution. And we saw that for both sea bass and sea bream, around 90% of the SNPs fell into this category. And that's actually at least one of the top performing SNP arrays in all aquaculture species. A very good SNP array performance, which was very encouraging. We'll hear a bit today about how the SNP array has been used in the MedAid project, but one thing we did as part of our study was look at the array performance in diverse populations. We did an initial test of the utility of the array for detecting population structure, but we'll hear from Beatrice later about more extensive use of the SNP array for looking at some of the population genetics of bass and bream populations in the Mediterranean. And basically our results there, the principal components analysis plots, show that we were able to separate out at least some of the populations as would be expected from the use of genome-wide markers to detect population structure. So beginning to wrap up, the combined species 60K SNP array was successfully created in the project. It was validated in multiple sea bass and sea bream populations. We had a database of tens of millions of discovered SNPs, which is a useful resource in itself. We then converted to the 60K array and had a 90% conversion from the candidate SNPs to the functioning SNPs on the array. The array was shown to be informative for diverse farmed and wild populations across the Mediterranean. We showed that it's useful for detecting population structure, and we've made all the sequencing data and indeed the access to the SNP array completely open access for use by the community, scientific or industry. So how's that having impacts and how's it being exploited? Well, we'll hear today or have heard today about some of that. So downstream activities in the MedAid project that includes looking at the genomic analysis of production traits led by Nofima, the estimation of population relatedness and loss of genetic variation, INEA, and G/E interaction, which we just heard from John there. More broadly, the impacts for the industry, well, we're already seeing companies purchasing the array, which leads me to believe that it will be widely used. It will be an enabling tool for the application of genomic selection technology as the industry starts to uptake genomics more routinely in their breeding programs, and I think we're definitely starting to see that happen, but more generally just a unified platform for consistency and cross-study comparison for both academic studies and for industry scientists as well. You can read more about this work, which was recently published in the journal Genomics, and I'd just like to particularly acknowledge Carolina, who did the bulk of, or a lot of this work that I've presented today. But as I said, it was a great collaboration with PerformFish and Teresa and Costas in particular were leading authors of this work as well. So that's it, and I'd just like to thank a lot of people, my co-authors, as well as all the companies and partners that kindly provided samples, and of course the coordinators of the MedAid project.